Hello, in this video we will discuss about the protein. Basically we will understand about what is a protein. So in our body basically 62% is water and 18% protein and 16% fat, as well as the carbon is the carbs is 1%. So here is the body composition with the macromolecule. So let's begin to understand the macromolecules, the protein. This protein is made up of amino acid chain as a unit of protein, and different protein play different function. So let's begin to understand. So amino acid in our body is 21 to 23 total amino acid in body. So the amino acid is organic compound containing amino and carboxyl group. So this is the basically different protein play different function. For example, hemoglobin, which that is used for the oxygen and carbon dioxide exchange. So the amino acid in our body is basically 21 total or 23. So these all amino acid composed to get a new sequence of the amino acids known as the polypeptide. That's polypeptide is play important function differently. For example, the different protein play important role for their different function in our body. So the amino acid is organic compound containing amino and carboxyl group is here, and here is the side chain for different amino acid. Basically the carboxyl group is here, and here is the amino group, which that is the amine is attached here. So let's begin to understand the basically zwitterion. Basically zwitterion is the two types of the transition of the two type of amino acid. For example, here is the neutral structure, but the shifting of the proton toward the amine group is create a positive and negative charge. So it means it is amino acid basically is not acid, but it is dual nature, and there due to the zwitterion. So the dipolar structure is also called internal salt. So basically, for example, it is show the dipolar. Why? It is like a sodium chloride salt, which that is the sodium chloride contain a negative and positive charge. So this is also play important role as a salt. So that is all amino acids exist largely in dipolar form. For example, acid and base. Let's begin to understand the analysis of the acid and base. So when we will put the addition of acid, we will add the acid into the amino acid. So it means the positive and negative charge, basically negative charge will attract the proton. After the attraction of the proton to reduce in the after the accept, the proton will reduce into the positive charge. So that's it create the basic acidic character, because basic character, because it is accept the proton, while it can be lose the proton as a acidic character. When we will add the alkaline solution into the amino acid, so it means it contain a positive and negative charge, so it will be donate the negative charge to the alkaline solution, for example the hydroxyl group, to reduced in the form of negative charge, so release the water after the binding with the proton with the water. So that is important things. So here is a test of amino acid we will discuss. We can test the amino acid in the laboratory with the ninhydrin test. When we will add the ninhydrin solution into the amino acid to show the bluish violet color. So that is the identification by paper chromatography. So the next is we will discuss about the peptides of the protein, basically the peptide bond formation, which that is lead to polypeptide formation and produce the protein with the combination of the amino acids. So let's begin to understand. Here is the two amino acids when come together. So hydroxyl group of the carboxyls and the amine and the nitrogen end, which that is interact with a positive and negative charge to release the water, and after the water releasing is the condensation formation and release the dehydration will be occur, and the N terminus and carboxy terminus will be occur, which that is join another amino acid, and this bond is known as the CN bond, and this is also known as the peptide bond CN. So this is the basically the long polypeptide chain will be occur during the translation of the protein in our body. So we will also understand the translation of the protein in our body with a small overview. So the types of amino acid, first of all we will understand. Amino acids type is two types: non-essential amino acid and essential amino acid. Non-essential amino acid in our body, which that is produced by itself by metabolism, for example 9 amino acid, while the essential amino acid is produced due to the food, when we will eat some food, proteinaceous food, for example meat and egg, and rest of the amino acid are present by the dietary food, dietary protein. So the synthesis of DNA, synthesis of peptides in our body through the DNA. So the DNA contains gene, which that is specific gene produce the specific sequence of the polypeptide. First of all, the DNA will be transcribed into the messenger RNA, and that will be go into the cytoplasm and will begin the translation by the binding of the ribosome, as well as the transfer RNA. After the binding to increase the polypeptide formation, and the polypeptide bond will be occur. So that means the polypeptide will be formed, and after the modification in the rough endoplasmic reticulum as well as the Golgi bodies to produce a mature protein, which that is the shape different and they are structurally and functionally different, and they play important role in the different function. So let's begin to understand the structure of the protein. Basically, first of all, the primary protein. Primary protein is begin to produce during the translation to produce the polypeptide, the long chain, and this chain is the CN bond will be occur, the CN bond and the polypeptide bond, peptide bond. Sorry. So this primary structure, and after this primary structure will add the disulfide bond or another bond to form a secondary structure, and the secondary structure is the basically helical structure show. But important thing is that the secondary structure is the alpha helix structure. So basically the alpha helix structure is two polypeptide come together to form a disulfide bond, or hydrogen bonding, and the interaction of the hydrogen bonding are another hydrophobic and hydrophilic interaction to produce a shape of the protein, which that is the tertiary structure. So let's begin to understand the tertiary structure. Tertiary structure is produced due to the intermolecular forces as well as the hydrophilic and hydrophobic part of the protein, which that give the shape, and this shape of the protein will lead to produce a phenotype from the genotype. So that is a phenotype, because the phenotype is due to the appearance of the translation of the gene. So that is the translation of the gene. And the tertiary structure is also produce the quaternary structure by the binding of the more than one tertiary structure protein with the aggregation of more than one polypeptide. So here is a hydrogen bonding. This hydrogen bonding is basically due to the hydrogen, and covalent bonding can be occur in the tertiary structure as well as the polar interaction. So that is produce the three-dimensional structure. We will also understand about that. So first of all, here is the quaternary structure, which that is made up of aggregation of more than one polypeptide. So the more than one polypeptide of the tertiary structure will lead to produce the quaternary structure. So let's begin to understand the three-dimensional protein, which that is used as a function. For example, the hemoglobin, which that is used for the respiration, oxygen dissociation and association as well as the carbon dioxide, and hormones, which that is insulin, for example, is signaling molecule, and albumin, which that is used for the osmolality in the blood, and enzyme is used for the metabolism, and actin, tubulin, and keratin is used for the structure, muscle cell hairs. So here is the lungs. Let's begin to understand. For the expiration and inspiration of the carbon dioxide and oxygen is basic rule for the attachment due to the iron associated protein is known as the hemoglobin, is used for the transport, while here is the hormone is used for the insulin produced, for example, to use as a signaling molecule and to lower the blood glucose in our body, while the albumin is basically used for the osmolality. Sodium and potassium concentration remain constant, as well as the water concentration homeostasis. It is used for the transport, while the enzyme is basically used for the metabolism, for example, anabolism and catabolism. Let's begin to understand with the example of the enzyme. For example, enzyme is basically pepsin, which that is used for the breakdown of the protein. It is known as the catabolism, while the metabolism is basically anabolism is the synthesis of the polypeptide or synthesis of the another things. So the peptidase enzyme or another enzyme, which that is used for the catalyzation, and that is known as the catabolism. So here is the central dogma for the production of the protein we will discuss here. So here is a cell which that is contain a DNA. DNA contain a specific gene which that is produce the specific messenger RNA. This is known as the transcription, and messenger RNA will go into the cytoplasm with bind with the ribosome, as well as transfer RNA. One transfer RNA contain a one amino acid. So the multiple transfer RNA will be bind with the codon by the anticodon, which that is transcribed the one amino acid. So that is produced a chain of amino acid. So this chain of amino acid is known as a translation to produce protein. This protein, after the modification, will become a part of body, and that's show the phenotype of the genotype. So that was the protein. Basically, the protein is the basic contain a amino acid, functional and structural unit of the body. So I hope you make sense about the protein concept. So thanks.